Great, thank you. I really appreciate the opportunity to be here. I appreciate y'all being here. I wanted to share some of our recent data that we've been generating. As Maggie said, this is focusing on ALS, FTD, Parkinson's disease, and Alzheimer's. My primary interests are in Alzheimer's disease and ALS and FTD, but we wanted to focus in on some regions of the genome that are particularly challenging to resolve, that they cannot be resolved with short-read sequencing data. They can be particularly challenging even for standard long-read sequencing. So first, I just want to briefly tell you why optical DNA mapping. So even a year ago, I was not using optical DNA mapping, and I was a bit skeptical about using it. In talking with various people and also looking at the data myself, we've been convinced of the incredible value that it brings. And let me tell you why that is. So first, the Saphyr, I was a slide behind. No conflicts of interest. Here we go. Okay, so with the Saphyr, it provides unparalleled N50s. If you're not familiar with this metric, it's simply a measurement of how long the molecules are. And you can also use that same measurement for the assembled contigs based on this data. It also has really deep coverage, and all this at a very affordable price. So with the N50s, because the molecule lengths are so large, you can resolve both haplotypes for an individual for some of the most challenging regions in the genome. With the coverage, you get 100 to 400x coverage, and this allows you to assess mosaicism or to multiplex. And it's all quite affordable, so you can do large cohorts. And this was one of the big things where bringing all those three things together, you can't find that with any other technology right now. So some additional features that I was actually quite impressed with that I didn't know any of this before we even started doing it. Being a bioinformatician, I'm usually quite comfortable getting into new technologies because I can do the work, and most of the other technologies, they require that work. But I was really impressed at how automated and mature the bioinformatics process is with Bionano. Let's see here. So I also, I guess I want to show some of the data that we've been generating. So we performed optical DNA mapping because we wanted to look at some of the challenging regions. And this is across 31 brain samples, all from the frontal lobe. Some of the regions that we're going to focus on are the ALS FTD causing C9orf72 G4C2 repeat expansion, GGGGCC repeat expansion. So if you're not familiar with this, it's in the gene C9orf72. The repeats can become extremely large, up well above 50 kb even, and it causes ALS and FTD. We had a sample with a known alpha-synuclein triplication, but we did not know the genomic architecture of that. We're also going to take a peek at CR1, which is a top Alzheimer's disease gene, and that's become a particular interest for my group, and I'll tell you a little bit about that. And then MAPT, or the tau protein. This is a hallmark pathology for Alzheimer's disease and various other tauopathies. It's a really important player, and it also has a known inversion that you cannot resolve with short-read sequencing data. And then finally, the Histocompatibility Complex. This is a very large region that houses the human leukocyte antigen genes involved in immune system, and it's been implicated in over 100 different diseases. But no one has been able to really resolve why. And I think it has a lot to do with the genomic architecture, because it's actually quite complex. So first, ALS and FTD. As I mentioned, the G4C2 repeat expansion causes that. On the right, we're showing a southern blot where we've done, the southern is from 3 different brain regions, the frontal, the parietal, the temporal, and the cerebellum. And this repeat expansion it demonstrates mosaicism that you can see even by southern blots within tissue, so a single piece of tissue. These are not like adjacent pieces, a single piece of tissue, and then across the different brain regions. So here you can see the band is a smear, and that's demonstrating that there are different lengths that they're at least suggesting that there are different lengths. And then compare the frontal and the temporal compared to the cerebellum. The cerebellum is always shorter than the other regions, which I think is quite interesting. So we picked a sample. We wanted to pick the hardest sample we could to try and challenge, the Saphyr. So this sample, it's a bit faint, but there's a band up here that's greater than 23 kb. Then there's a small band here, and then down here, this represents the reference allele, which is usually either 2 or 8 repeats. So in this case, there are three. We would expect 3 primary populations of repeat expansion sizes, and this is quite rare. Most of them are only 2, as you can see here. There's usually the expanded allele, which could be anywhere in this gradient, and then the reference allele. So we were really impressed to find that using the Saphyr, we were able to assemble or resolve these different lengths. So if you're not familiar with this, so this is the gene CNR72 on chromosome 9, and this represents, the green represents the chromosome 9 of the reference genome. And then each blue line represents an assembled haplotype. So in this case, we have 5 distinct haplotypes, most of which fall within this larger band. But it gets all the way down to the 1.9 kb insertion, which is about right if you take into account flanking sequence. But you can see this progression where each hash here represents the motif that is tagged by the Saphyr, and this is where you would expect them in the reference genome. And you can see the alignment is perfect for each haplotype up until you get to the region where the expansion exists, and you can see this natural continuum of the repeat size. And looking a little bit more closely at this region, this is the same sample where we're showing the assembled haplotype with no repeat expansion, and then this is an assembled haplotype averaging all of these rows below, where each of these is an individual DNA molecule that has been mapped, which I think is part of what makes this so valuable. The first thing is I really want to emphasize you can construct both haplotypes, both germline haplotypes, and in the case of mosaicism, we constructed 5 haplotypes for this expansion. It's really, really remarkable. But on top of that, you can investigate individual molecules, and that's really fantastic. So this largest one is 45 kb. And that should give you an idea of just how big these molecules are, because with other technologies, while they have a lot of value, they're fantastic, you're really unlikely to get even a few reads, or you'll get a few reads that reach 45 kb. You're not going to get any reads that can span a 45 kb expansion. It's really impressive. So turning to the alpha-synuclein example. So again, we knew that there was a triplication. This individual had 4 copies. It's implicated in Parkinson's disease. But we didn't know the haplotype structure. So right off the bat, using the Saphyr, it is able to identify that there are 4 copies, and the region. Like, we didn't know how much of the region had been duplicated. So it's about a 2 megabase region that's been duplicated. If you start to look, if you look down at the gene level, again, we have the assembled normal allele with no duplication. So now we know that the triplication is on 1 haplotype. Instead of being 2 and 2, it's 1 and 3. And we're able to identify that 2 of the copies are actually inverted. So 1 is the standard direction, and then the other 2 are actually inverted. So again, we're able to resolve that structure, which is really important, and important proof of principle that we can capture these complex rearrangements. In NCR1, I told you this is a gene that's of particular interest to my group. we published a paper a few months ago on the dark and camouflaged genome. I don't know if you've seen that paper. I personally like it. But as part of that paper, basically we identified a lot of genes in the genome that are completely overlooked, or at least there are regions of the gene that are completely overlooked based on short-read sequencing data. So you're sequencing, your cohorts, thinking you're getting all the mutations for all your favorite genes, and you may not be. And I was surprised, because I've been in Alzheimer's disease research for over 7 years, to find that one of my favorite genes, CR1, is a top 5 Alzheimer's disease gene, and it's 26% camouflaged. And I won't get into what camouflage means at the moment, but here I'm showing a coverage plot of the NOMAD consortium. If you're not familiar with the NOMAD consortium, it's an aggregate of 140,000 whole exomes and whole genomes. So this is showing that in all of their data, this whole region, these regions are completely missed in all short-read data. So that was a surprise to me, as I mentioned. So what makes it even more interesting is that the region that is camouflaged is the C3b/C4b binding domain, which helps regulate the complement cascade process that's implicated in Alzheimer's disease. But then even more, the number of times that this binding domain, so in HG38 it's repeated 3 times. This binding domain, the number of times it's repeated is actually variable in the population. So we all have different combinations of how many of these repeats are actually in our CR1 gene. So, and then just to add another layer of coolness, I think, to this gene is the number of times this domain is repeated is actually associated with Alzheimer's disease. So you cannot construct the haplotypes for this with short-read data. You can't do it. And it doesn't work. It doesn't seem to be working super well with long-read data either. But Bionano nails it, and I just want to show you an example here. So again, this is just showing the reference chromosome 1, and I'm only showing the haplotype. This individual, they had a normal haplotype with no deletion, and then the second haplotype has a deletion of 1 binding domain, and you can see it quite clearly here. Just really impressive. So now we can directly measure the number of times that this binding domain is measured and perform more precise, more accurate association studies to determine if this is in fact driving Alzheimer's disease, or at least modifying. So now turning to the MAPT or tau, as I mentioned, this is a hallmark pathology for Alzheimer's disease and other tauopathies. And there's a known inversion of this region that involves MAPT. So this is the H1 haplotype, and you can see the directionality of these genes that are involved, where MAPT is here. And then the H2 haplotype is this whole region is inverted. Again, you cannot resolve this structure with short-read data. It also turns out that the H2 haplotype is associated as a protective factor for various tauopathies, including Alzheimer's disease. With Bionano, had no trouble resolving that. So we have haplotype 1 with no inversion. Haplotype 2, again, I just think this is really great. We're not just looking at, with standard sequencing, you look at a position, they have an A and a T, but you don't know, does this T go with the mutation upstream? You can't do that very well with short-read data. With this, you can. We have the whole haplotype for this region. And so we were able to identify these inversion breakpoints, but what we did not expect was that in one of the individuals with this inversion, not all of them, but they had de novo sequence that does not align anywhere in HG38. And that could be an important disease modifier. So this was a big surprise to us, but really cool. And then finally, to the Histocompatibility Complex. As I mentioned, it houses the HLA genes, and I've tagged a couple of them. It's a large region. It's about 4 megabases, but hopefully to help orient you, the HLA DRB5 or HLA DRB1, I think, or DR1, I can't remember. That region is strongly implicated in Alzheimer's disease, and as I mentioned, overall this region is associated with over 100 diseases. But no one's been able to resolve this structure But we're able to resolve the entire region, a 4 megabase region. And we did this across all 31 samples, as I mentioned, and you can see distinct haplotypes for most of the individuals. So here and most of them are at the HLA-A and the HLA-DRBA5 regions. Most of them, there are a few others sometimes. So most of the genomic variability is in these regions, and you can see just based on the banding pattern of sequence that does not align anywhere to the reference genome, and that's just indicating variability, and it's different for most of the individuals, which you would expect. So in conclusion, I've just been really impressed with what we've been able to do with the Saphyr. It provides genome-wide structural variants for, again, both haplotypes. To me, that is a big deal because we're no longer just associating an individual mutation. We can begin constructing an entire haplotype and testing that association. It's affordable and amenable to both clinical and environments and large cohorts. So I spent a fair amount of time in industry at ARUP laboratories helping develop clinical diagnostics, and so I'm familiar with the importance for low costs. And now being in academia, I'm even more familiar with the importance of low cost because I have to try to come up with all my own money to do everything. So, but that was one of the things that really got me again is the depth, the read length, and the price. You can do large cohorts to identify these structural mutations, and then the complex haplotypes. But with that, I'll stop, and I appreciate again your time. Thank you.